Okay, see that? Okay, that's a little bit better. It was a lie. It told me it was half lights, but clearly that seemed a little bit more than half to me. I don't know about you guys. All right, let's turn this mic volume down slightly because that is echoey. All right, much better. All right, great. So we're going to talk about the lab practical and the lab dog and how to succeed and not fail, which is important. So there's only 2 lab practicals tests, and they're worth very large chunkier grades. So it is important that you guys prepare for these. Basically, anything that we've covered since the beginning of the semester is fair game. Lab art isn't really going to be included in that, so don't worry about that. But that's all going to be fair game. So what should you do to succeed and not fail? Well, you could review the previous lab lecture PowerPoints, panic moments before the practical, write down C for every answer, and then don't put your name on it to delay the inevitable. That's my preferred method because it'll take me a while to go through that. A more realistic approach is still review the lab practical lecture PowerPoints. Those are still up. Those are still very effective. Know the steps in each test we do. So everything that we have done in lab has steps. You should know why we do those steps. So, for example, gram staining, you're like, oh, well, first thing we do is heat fix it. Well, why? Why do we heat fix things? Okay, I hear a lot of answers. Which is good. There's about 2 or 3 reasons why we heat fix things. One is to make them stay stuck to the slide, because if you just let them air dry, they just wash off the first rinse. The other one is to kill the bacteria so we don't have a biological hazard on a slide constantly. And the other one is it helps them long-term prevent autolysis, because if they just kind of sit there for a really long time, they'll start lysing themselves because their enzymes aren't inactivated, so heating them tends to inactivate them. Okay, other example is like, hey, what do the different, so knowing the reagents we use and why we use them, and it's, maybe reagents isn't the best word, but like, so knowing nitrate reagent A and reagent B and zinc and why we do them and what order we do them in and what those results mean. Knowing what we use in an EMB plate or a Conkey's auger plate or an MSA plate and what do those things look like. And the last one is to know what those results look like, which is actually probably the most important. Man, the computer is thinking really hard lately. Okay, knowing what those results look like, because it's especially the last 2 labs kind of threw a ton of information at you guys. Those are probably the biggest hurdle to overcome, so make sure you guys are, really spend some time learning those. If you're not sure how one of them works, make sure to ask, especially me or your TAs. So we're going to talk about that in just a second. So what is it going to look like? Well, Monday, Tuesday, we're going to do something called a skills check and then the TA review. We'll do 2 skill checks. Sorry, you guys aren't Tuesday, obviously, but there's another class that's going to do skill checks on Tuesday. They have you guys are going to do a streak for isolation plate. So we're going to give you a sample, it's in broth. You're going to do a streak plate, streak for isolation. Key thing for this, you have to A, make sure it doesn't have any contamination on it. B, make sure that you do a correct streaking pattern. C, incubated properly with correct label. This is the big one, because your TA is going to grade them, and if they can't figure out whose it is, you just get a 0, which is not good. So don't do that. Don't make them give you zeros. The other thing that's actually not listed on here, but is important, is make sure you tape them up individually. And the reason why we tape them up is because if they get knocked over in there, it's like 100 plates, or it's not 100, but like 25, 30, 40, 50 plates, depending on which incubator you're using. They get knocked over, and now you've got a giant biological mess that's spilled everywhere. So tape them up individually, because we're not doing it as a group, but tape them up individually. And your TA will check to make sure you've got an isolated colony. That means it's not touching anything else, it's just by itself, and it is the right kind of bacteria. It's not some other random bacteria. Lastly, you will be doing something called a pipetting volume check. So we will give you a specific volume, and you will have to set a pipette. You're going to have to choose a pipette, set it, the right volume, show your TA that it's the right volume, and then you're going to pipette liquid from one container to a microtube, and then we're going to check to make sure it's the right volume. On top of this, and it's really important to make sure that you guys maintain sterility in this. I know it's not quite something we've harped on a lot, but this is actually really important. Making sure you open the lid box, grab the tip, and close the lid box right away. Don't just leave it open, because then it's contaminating things. Don't then touch the tip with your hand or on the countertop or on the outside of the tube, because that's not a good idea to do, because that contaminates it. So please don't do that. Make sure that you guys are thinking through that. And of course, setting the right volume and the right, choosing the right pipette. Yes, a P1000 could pipette 20 microliters, but it's not a good idea. It's not going to be accurate. Don't use the P1000 for that. Don't use the P200 for that. Use the P20 for that. Okay. After that, on Wednesday for you guys will come in, you'll sit down, and we'll have the normal lab practical. There's going to be 24 stations. You'll go to each station and answer the questions on it. They are fill in the blank, so you got to really know your stuff. You can't just guess your stuff. Well, I mean, you could guess, but you can't just hope that there'll be answers there. Everybody will have 2 minutes per station. So you'll sit down where you normally sit down, start working on it, then scooch over one after the 2 minutes and start working on the next one. You guys will do this 24 times, and then after that, at the end of the class, if there's time left over, and this is where it's really important that you guys start class right away, so don't filter in 5 minutes late. We'll give you guys time to go and review any other stations that you're like, hey, I'd really like to check back this one. I can't remember station 13. I can't remember if this was a gram positive or gram negative. I forgot completely. I didn't write it down. With that being said, though, you guys should make an effort to A, have your papers closed up so that people can't see what you've written on your answer keys when you're moving around and viewing other stations, and not to be stacked up on top of each other, all looking at the same station. So if there's 4 people on the same station, that's not very helpful. So make a line that's a little farther back so you guys can do that. Your TA will shoo you away, but make sure that you guys kind of set that up. So the midterm, historically, has kind of taken a big hit in terms of grade because people are unprepared for it. So I will scare you a little bit by saying that so that you guys will make sure to put that effort in to study it. Be prepared. So we're going to give you guys a couple of example tests. Hopefully you guys don't have your slides open so you can't see the answers, but we're going to talk about these questions real quick. Okay. So, give you guys a slide. Go, hey, what is this? What kind of stain is this? Acid fast, very good. What's the primary stain? Carbonyl fusion's right. I remember that. Why do we use steam? To penetrate the mycolic acid. Very good. All right. What is the result for the bacteria that the arrow is pointing to? Non-acid fast. Very good. Acid fast negative is another way. We'd both take that answer. Okay, and by the way, I'm not going to be a stickler for spelling because I didn't get into biology to spell things correctly. I use computers for that. But try to make it really close to the word so we can figure it out, okay? Also, small differences in okay, you said non-acid fast. It says it's acid fast negative on the key. That's fine. We're not going to quibble over that. Okay, good. Real quick, so this is a bonus question for you guys. Does anybody here know why mycolic acid resists staining? Charge. What about the charge? What was that? No. Mycolic acid has a property to it. What is its property relating to? It's waxy, but what is its property relating to charge? It's wax. Yeah, okay, you got it. So oils and waxes are typically non-polar, hydrophobic. So they're not going to want to have charged things coming up against them. And because dyes are actually charged, generally speaking, they're going to resist staining. And the way we get around this is with heat, which helps move apart and make it less picky about what's going into it. But then that also allows our dye, which is slightly non-polar, carbonyl fusion, which has a phenol group on it, allows it to penetrate easier into the cell. Okay, good. All right, next one up. Here's a plate. What kind of plate? Blood, very good. What kind of organismal growth does it support? Anything? Fastidious is the word I was looking for. Fastidious organisms. It supports the growth of lots of organisms, but it's fastidious that it supports because those guys are picky, right? All right. What is the result of this plate? Beta. Okay, wow, I am impressed that there is such clashing of answers here. This one is actually gamma, or non-hemolytic. It's the same thing. And the reason why, if you guys look at this, you can see the colonies on here are white, but you cannot see a clear zone behind them. The bacteria are just sitting there on top. The area around them is not clear or brown. When you guys actually see this in lab, though, they're going to be sealed plates. By the way, don't open up any of the plates, please. But they'll be sealed plates, so you can pick them up and look at them, hold them up to the light if you need to like visualize them a little bit more clearly. Totally fine to do. But there you go. So this one's gamma, non-hemolytic. Either one would work. All right, what makes this plate opaque? Blood, a little bit more specific. Sheep blood, a little bit more specific. Red blood cells, yes. Or erythrocytes, if you want to get technical. Because blood itself is kind of like a loose term. It's like a bunch of stuff in blood, right? There's like different types of cells and platelets and nutrients. But like blood itself is actually clear, right? We can spin it out and it'll be clear on there, right? So don't just write blood. That's not a good answer. Okay. All right, things to study. I know it seems scary when I write it out like this, but remember, these are like, some of these are like doubled labs, like we did them on the same day, okay? So first one that's really important that you guys might neglect because you're like, hey, I already know this stuff, is lab safety. Make sure you go over lab safety protocols. Know what proper protective equipment is, know what proper lab attire is, know where the safety stuff is. Hey, where's the chemical safety shower? Kind of important. Should you drink food in lab only when the teacher's not looking, or never? That's actually the better answer. Okay, but aside from that, more importantly is basic microscopy, knowing how to do basic techniques, what all the parts of the microscope are, things like what is resolving power or working distance, also being able to operate a microscope because we will have you guys bring things into focus. So make sure you guys do that. Make sure that you guys spend time learning that skill. If you guys don't, you guys are a little rusty on your microscopy skills, guess what? Next week on Monday, good time to practice. Be like, hey, can I borrow the microscope for a bit and use it at my station and practice? It's a good idea. All right, next one up is pipetting. Obviously that's a skills check, but we're gonna have some questions on pipetting. Gram staining, know all the procedures, know all the techniques, know how to interpret results, know the difference between cocci, bacilli, strepto, staphylo, random arrangements. Endospore acid fast staining, you should be able to look at those and go, yeah, that's acid fast, that's non-acid fast, that's an endospore, that's vegetative cell. Negative staining, that's a negative stain. These are the chemicals that we use for a negative stain. This is what this result is. Transformation, we'll have a transformation plate potentially, or something else, and ask you some questions about that. Also, you should know the kind of steps that we go through for the transformation. Like, hey, how hot do we have to make it to do the transformation? What is the incubation temperature that we do it at for the brief heat shock period? You should know that. You should know what temperature we incubate them at. Things like that. All right. Culture techniques and isolation, basically how to inoculate things properly. It's not going to be a huge thing because there was a ton of material in there, but it's proper inoculation techniques, knowing what tools to use when. Viable plate count, knowing how to count up viable plates and do the equation for it and figuring out how many bacteria per mil are in the original sample. That will be a whole question. And then there might be some other questions involving that, so keep that in mind. Oxygen requirements, BHI auger deeps, looking at them going, hey, this is a microaerophile, or this is a strict anaerobe, things like that. Water testing, we just did the water testing, same kind of thing. Make sure you know how to read an MPN chart. Make sure you guys know how to interpret the EC mug things. Make sure you guys understand what the water testing involves. Selective and differential media, that was just recently. It's got a lot of stuff in it. Make sure you guys run through it. And of course, the nitrogen cycle. Bonus questions. So we will have about, I think I've got it at 5 or 6 bonus questions. Each bonus question is worth 1 point. So the total number of points on this exam, I think, is 75. So 5 points is a huge chunk of your grade, right? So if you guys spend a little bit of extra time knowing those, then you guys will be good. Well, you guys will potentially get some bonus points. Now, here's the thing. Bonus questions tend to make it a little hard because I expect them to be difficult to get. But they're going to be on the cool science thing of the week. And I only talked so much about it. And if you guys paid really close attention, I tended to tell you what the biggest thing to take away from it is. Maybe. That's probably a good idea of what I'm having you guys look at. Okay. Okay. Format stuff. So, short battle royale will occur at the beginning. You can slaughter out your classmates and take their points. Cool idea, I know. Really pushing the forward of education. No, we're not going to do that. Okay. We'll have a total of 25 stations. The 25th station will be the bonus one. You don't have to do it, so you can just sit there and ignore the bonus one, but you can go through all the other ones. You'll have 2 minutes per station. Once the time's up, we're going to make you move, whether you're ready to or not. Remember, you can go back and review it if you got time left over. Make sure you guys don't form a line. Any questions? Use Google? No. Does anybody have any questions concerning this? Oh, and I should say one last thing. Assuming I have my files. If on Canvas there is a little deal that looks like this that says Lab Practical Review document, and it's kind of got a bunch of fill-in-the-blank stuff. So you guys can test your ability. It's not on there. Okay, hold on. There we go. Thank you. Ignore this one, though. You obviously aren't doing that. But everything else you guys are doing. So roll through that, and if you guys have questions, that might be a good time to bring them up with your TAs. All right, any questions? Yes. It's all fill in the blank. No. Shh, guys, quiet down so I can hear the question. Monday, the skill check is part of the lab practical, so that's just getting added on top. Any other questions? Yes. Bonus are going to be fill in the blank too. Any other questions? Huh? You can ask questions on Monday. Then we get docked points like a Huh? What? No, you won't get docked points. You can just ask questions to the TA. It's a review day. Skill check will happen at the beginning, and then after the skills check is done, then you guys can ask them questions. All right, guys, have a safe weekend. I'll see you later. Good luck studying, and shoot me an email if you guys have any questions.